Good evening, students of medicine and health sciences everywhere, especially my dear students. This is Dr. Akra is greeting you and ready to start with you an interesting lecture regarding the gas transport. We mean by gas transport one of the important step in the respiratory process in our organism. Here we are talking about the blood gases. I mean the oxygen and carbon dioxide. Okay, actually the transport is transporting of oxygen from the lung tissue toward the peripheral tissue where the metabolic processes are active. And then the transport of carbon dioxide, CO2, from the peripheral tissue toward the lung tissue where through the process of gas exchange would be exhaled by the lung out of the body toward the external atmosphere. Okay, first of all, there is a difference between the transport of oxygen and transport of carbon dioxide. How? As you see, oxygen is not dissolved, is not transported as dissolved gas in the arterial blood. It is transported only bound to hemoglobin. You see these buses full of hemoglobin passengers who were waiting here in the lung, I mean in the alveoli, before exchanging with the capillary blood, then transporting toward the left atrium and left ventricles as oxygenated blood, and then toward the peripheral tissue. Actually, hemoglobin has two components. The proteinic component, which is globin, composed of 4 subunits, 2 alpha subunits and 2 beta subunits. Each of proteinic subunits of hemoglobin actually involves a non-proteinic component, which is the heme. For that, the 4 subunits of hemoglobin actually involves 4 heme groups. Each group is bound to iron, but the iron is it is reduced form, not it is oxidized form, because the oxidized form actually is called methemoglobin. This methemoglobin, actually the iron lack an electron which is necessary for binding oxygen. Okay, now each iron in the hemoglobin actually bind to one molecule of O2. So one hemoglobin plus 4 O2 will lead to formation of this. And binding of hemoglobin with oxygen will increase the affinity of hemoglobin to bind to oxygen. This is a reversible reaction, as we will see, because here the oxygen or the hemoglobin will unload its content of oxygen. So when the affinity is increased, this will encourage the hemoglobin to bind with more and more O2, and so on. Here we have 8 atoms of oxygen, which means 4 molecules of oxygen. At this level, the hemoglobin is totally saturated by oxygen. Okay, about 97.5% of hemoglobin in the blood is actually saturated with oxygen, while only 1 to 2% oxygen is transported as dissolved gas. For that, the transport of oxygen as dissolved gas is not necessary at all to sustain the needs of the peripheral tissue. In each 100 milliliter of in each 100 milliliter of arterial blood, there is only 0.3 milliliter of oxygen in the solid form. For that, the vast majority of oxygen is transported bound to hemoglobin as oxyhemoglobin. Here, we have to distinguish between three important terms. The O2 content of the blood. This involves the dissolved O2, only 2%, as I mentioned, 1 to 2%, plus oxyhemoglobin, which is the oxygen bound to hemoglobin in the blood. While there is another term which is hemoglobin carrying capacity of oxygen. Now, in each milliliter of the blood, there is about 15 gram of oxygen. 15 gram of hemoglobin. 15 gram of hemoglobin in each 100 milliliter of blood. And each gram of hemoglobin is capable to carry 1.39 milliliter of oxygen. By multiplying 15 gram of hemoglobin with 1.539 milliliter of oxygen, the carrying capacity of hemoglobin will be around 20. So there are 20 milliliter of oxygen carried by the 15 gram of hemoglobin in each 100 milliliter of blood. And the hemoglobin saturation of O2, this is difference between the arterial and the venous blood. Why? Because the saturation is increased when the partial pressure of O2 is quite high, as in the lung tissue. In the arterial blood, as I mentioned, about 97% of O2, of hemoglobin, is saturated with oxygen. Now, if we have three persons, one who is healthy, the other has anemia, and the third has polycythemia. Actually, the oxygen content and the hemoglobin carrying capacity in both would be different, for sure. But the hemoglobin saturation will be the same for the three persons. Okay. After reaching the peripheral tissue, as I mentioned, the hemoglobin start to unload its load of oxygen. Why? There are different factors that affect that. As you know, in the peripheral tissue, there is an increased partial pressure of CO2 and decreased partial pressure of O2. When the partial pressure of O2 is decreased, this encourages the hemoglobin to dissociate with oxygen. And the oxygen will go out of hemoglobin to go to the peripheral tissues and to maintain the needs of the peripheral cells. And the other factor that will promote the oxygen, the hemoglobin to dissociate from oxygen is the increased partial pressure of CO2. This is called pore effect. I think that you have studied that in the biochemistry. Pore effect means when there is an increased partial pressure of CO2, the affinity of hemoglobin toward oxygen is decreased and the dissociation will occur. Okay, how the dissociation occur? As you see here, this is the well-known or the famous hemoglobin dissociation curve. Please pay attention here. When there is 100 millimeter mercury of partial pressure of O2, which is the maximum almost, there is around 100 or let's say 97% of hemoglobin saturation. This side of the curve actually represents the hemoglobin saturation of oxygen as percentage, and this side of the curve or this axis actually represents the partial pressure of O2. So, when the partial pressure of O2 is quite high, the saturation of hemoglobin is almost the maximum. The good news here, when the partial pressure of O2 start to decrease, the dissociation of hemoglobin would not be linear. Only at 60 millimeter mercury partial pressure of O2 decreasing will decrease the saturation down to only 90%. This is quite good and healthy and enable us to survive and adapt when there is a decrease in partial pressure of O2 as occurs in high altitude, for example. And almost half of the saturation occurs only when the partial pressure of O2 is decreased to 27 millimeter mercury partial pressure of O2. This only occurs in peripheral tissue when there is an extreme or strenuous exercise. Okay, but are there any factors that causes faster dissociation of O2 at higher value of partial pressure of O2? Actually, yes. And this is called the shifting to the right. Shifting to the right means that the hemoglobin dissociates from oxygen at higher level of partial pressure of O2. This occurs when there are decreased partial pressure of O2 as we mentioned, and when there is an increase in partial pressure of CO2, when there is a decreased pH, I mean in acidic blood, and when there is an increase of what is known as diphosphoglycerate. Diphosphoglycerate is actually a metabolic end product of the RBCs. When it is increased, this will also causes more rapid dissociation and shifting of hemoglobin curve to the right. Also increased temperature will causes this shifting to the right. As you see here, increased partial pressure of CO2, decreased pH, increased temperature, increased diphosphoglycerate, all of these are actually, especially these three factors, all of these are actually markers of increased metabolic activity in the peripheral tissue. Okay, now let's go back to the figure here and to have a look at the transport of CO2 toward the lung from the peripheral tissue. As you see here, the buses are still having red passenger or still having oxygen, because as we mentioned here, only 50% of saturation is affected when there is very low partial pressure of O2 in the peripheral tissue. Now, the CO2 is actually transported in three ways from the peripheral tissue to the lung. About 10%, the walker here, are sign of the dissolved CO2. 10% of CO2, almost 10% of CO2, is transported as dissolved carbonic dioxide. Why? Because you remember from previous lectures that the solubility of CO2 is about 20 folds higher than the solubility of O2 in the blood. And about 30% of CO2 is transported in RBCs as carbaminoglobin. In hemoglobin actually, in RBCs and hemoglobin. And about 60% of CO2 is transported in these green buses, I mean as a symbol, which are the bicarbonate. So 60% of CO2 is transported as bicarbonate, 30% as carbaminoglobin, and 10% are dissolved as dissolved CO2. Now here we will describe two mechanisms related to carbon dioxide transport. The first one is the chloride shift. In the peripheral tissue, here you see the RBCs, which has a biconcave disc shape. The CO2 is diffused inside the RBC and combined to water. This will reveal the carbonic acid H2CO3, which is a product of the reaction that combines CO2 with the H2O. After that, the H2CO3 will further dissociate into the bicarbonate HCO3 minus and H plus. H plus will be trapped by hemoglobin. The HCO3 will efflux out of the cell in exchange with the chloride ions. After that, the RBC will be transported toward the lung, where the HCO3 will influx again inside the cell in exchange with the chloride, which will be effluxed out of the cell. The HCO3, which is the bicarbonate ion, of course, will combine with H plus to produce H2CO3 once again, and the H2CO3 will further dissociate into the CO2, which will be diffused out of the cell and of course will be exhaled out of the body, and the H2O, which will be either stay in the cytosol or under the osmotic effect will go out the RBC. This is called the chloride shift mechanism. Another mechanism, which is the Holden effect. Holden effect means that when the RBCs are in a medium with high partial pressure of O2, this will decrease its ability to carry CO2 and thus will release CO2 out of it. Thank you for being good listeners for today's lecture. See you.